This is Naftali Muhumza Medical Sciences, where we are taking you through a diagnostic technique known as xenodiagnosis. Xenodiagnosis, this is a technique, we are calling it a lab technique or method, which is being used whereby we use a vector, an insect vector, for example, a triatomine bug, for culturing. So we are using a vector as a culture media so that we multiply these organisms for easy examination. So meaning this method is used when the parastemia is low, the concentration of the parasites are low in the presence of triatomine bug. So the vector which is being used in the diagnosis of Chagas disease, so this one is being used to diagnose Chagas disease, or which we can call American Trypanosomiasis. And the vector being used is the triatomine bug, and we are using it as a culture media to multiply these trypomastigotes, which are suspected to be in man. So when do we do it? After knowing the significance, what we do, we starve this triatomine bug. It is a laboratory reared triatomine bug. You can call it a kissing bug or a reduvid bug. So this bug is starved for 2 weeks. After starving it for 2 weeks while being reared in the lab, we are going to allow it. This is our patient. And this patient, we are suspecting him to have what we call the trypomastigotes in his blood. So this is a man or a patient whom we are suspecting. So after starving this triatomine bug for 2 weeks, and we have to make sure this triatomine bug is uninfected. It must be sterile. It must be uninfected, in bracket, sterile, free from these trypomastigotes. So an infected laboratory reared triatomine bug is starved for 2 weeks. After starving for 2 weeks, it is allowed. We allow it to feed on patient's blood. This is a patient you are suspecting to be having Chagas disease, having trypomastigotes within his blood. So we are going to allow this uninfected starved laboratory triatomine bug to come and feed on this man. And when it feeds, it is going to take up his blood, which we are suspecting to contain trypomastigotes. So after feeding on this suspected man, the trypomastigotes are going to move to the midgut. So we are going to find the trypomastigotes. If the patient is having them, they move into the midgut. And within the midgut, they are going to undergo metamorphosis to form what we call the epimastigote. They form the epimastigote. Then these epimastigotes undergo division by binary fission, so they multiply in the midgut by binary fission. And we said we were suspecting the parasite to be low. So within the midgut of the triatomine bug, we are seeing the trypomastigotes metamorphosing into the epimastigotes, and epimastigotes undergoing multiplication to form many copies. And at the later stage, these epimastigotes, after multiplying and increasing in number, they transform again into the trypomastigotes, which migrate into the hind gut. So by the time we reach into the hind gut, the epimastigotes which have increased in number metamorphose again into trypomastigotes. So this life cycle in the bug, it is taking a duration between 25 to 30 days. So after feeding this kissing bug on the blood from the patient, we leave it in the lab for 30 days, for the maximum of 30 days. Even when we examine after 30 days, we can still leave it for another 30 days, every time examining for presence of trypomastigotes. So by the time these trypomastigotes reach in the hind gut, they have increased in number. So what happens, we are going to come and extract the fecal matter. So we come and extract, if this is the fecal matter within the hind gut, we come and dissect the bug and we extract feces from the rectum of the triatomine bug. So we get feces from the rectum of the triatomine bug, feces containing trypomastigotes. And it is these feces that we are going to examine. We come and examine them microscopically. Either we can use a direct wet mount, whereby you get these feces, you dilute it with normal saline, and examine for motile trypomastigotes. Or you can make a smear, you dry it, then we stain using Gimusa stain. Then we examine under times 100. So in summary, we have seen we are using the vector or an animal, an insect in the lab to act as culture media, for example, triatomine bug in the diagnosis of Chagas disease. And we are using an infected, starved triatomine bug or kissing bug, which has been starved for 2 weeks. Then after those 2 weeks, we allow it to feed on a patient which is suspected, to feed the blood. And after ingesting this blood, if it contains trypomastigotes, these trypomastigotes in the midgut, they metamorphose into the epimastigotes. And these epimastigotes multiply and increase in number by binary fission, and at the long run, they move into the hindgut, whereby they metamorphose again into the trypomastigotes. Then we are able to extract the feces of this triatomine bug, and we shall examine for presence of trypomastigotes, either using a wet mount. We can use a wet mount and look for the motile trypomastigotes, or we can use Gimusa stained method, stained smear, and we examine for presence. And how will they be looking like? We shall find the trypomastigotes. They are C-shaped, with the central nucleus. They have the kinetoplast together with the axoneme, then with a small undulating membrane. This is what we shall be looking for in a stained film with Gimusa stain. So this is what we can discuss with you under xenodiagnosis. Other methods for diagnosis of American trypomastigotes include direct wet mount for blood and the buffet coat, using Gimusa stained smear for the buffet coat, using serological tests such as ELISA, indirect fluorescence antibody test, and we can also do PCR. At the same time, we can also do xenodiagnosis. Thank you so much. May God Lord bless you. Please remember to like and share to many others.